Oxygen's role in our metabolism has been understood for centuries. We need it to survive, but for more reason than just making us energy. Oxygen is critical in one of life's most essential adaptive processes. Every instant, our bodies are sensing the level of oxygen in each and every one of our tissues. All of our cells are equipped with molecular machinery which can sense whether our tissues are hypoxic, meaning oxygen deficient, or normoxic, meaning having sufficient oxygen. After sensing the level of oxygen, our bodies respond, inducing changes in our metabolism and physiology. However, how our bodies actually accomplish this remains a mystery to many. How do we actually sense oxygen, and what kind of responses does it entail? The answer to this puzzle is not only essential to our understandings of our metabolisms and physiology, but also presents promising new strategies to fighting anemia, cancer, stroke, diabetes, and many other serious diseases. In hypoxia, our body responds by producing more of certain proteins such as vascular endothelial growth factor, erythropoietin, and induced nitric oxide synthase. These proteins come from a DNA sequence called hypoxia response element, and they help carry out vital tasks such as insulin secretion, red blood cell production, angiogenesis, and neural development. Let's take a deeper dive into this process. It all starts with a transcription factor called hypoxia-inducible factor 1. Its job is to convert the genes on the HRE DNA sequence into mRNA. HIF1 is made of two parts, a protein called HIF1 alpha and a protein called ARNT, or HIF1 beta. Under hypoxic conditions where there's deficient oxygen, HIF1 alpha binds to HIF1 beta as well as a transcription activator which helps it travel and bind to the HRE genetic sequence where it's able to transcribe mRNA. This mRNA then travels to a ribosome where it can be translated into our special proteins. In normoxic conditions where there's abundant oxygen, HIF1 alpha is modified through a process called prolyl hydroxylation, mediated by a protein called PHD. PHD converts oxygen into two hydroxyl groups which are added to HIF1 alpha. A new protein complex enters the scene. Its important parts are a protein called PVHL and another called ubiquitin. The PVHL constantly looks for hydroxyl groups, and when it finds one, it binds to the HIF1 alpha. A proteasome appears, hungry for ubiquitin. When it finds it, it uses enzymes to break down the HIF1 alpha. Because the HIF1 alpha is being continuously modified and destroyed, no new proteins can be made. All in all, the functions of the HRE proteins are increased under hypoxia and are inhibited under normoxia. Some of the functions which are increased include the growth of tumors, resistance to radiation therapy, heart failure, type 2 diabetes, neurodegeneration, and metabolic dysregulation. On the flip side, they also counteract anemia and are associated with cartilage repair and wound healing. By understanding the intricacies of this process, we can determine the root cause of such disorders. We can then develop treatments and preventative measures against them. We've already taken major steps forward through the study of this process. We're improving cancer treatment, curing anemia, repairing tissues, and even working towards anti-aging. We've learned so much, but we still have even more to explore in the world of oxygen sensing. And it's critical that we do, because in doing so we could find the breakthrough which saves countless lives. Thank you for watching.